Hi, my name is Fadi Lakkis. I'm the scientific director of the Starzl Transplantation Institute, and I'm also a transplant nephrologist. And I would like to welcome all of you to this event that we call the Research That Matters. This is an event that we do regularly at the Institute. And this year, of course, we're doing it virtually. And the goal of this event is to introduce our patients, their families, their friends, and the general audience to the research that we do here that is aimed at improving the lives of transplant recipients. In the next few minutes, I would like to give you an overview of why we do the research we do and what to expect during this virtual tour. So as all of you know, transplantation is a great success story. More than 100,000 transplants are performed each year worldwide for a variety of diseases, and they are life-saving. Organs such as the kidney, heart, liver, and lungs are transplanted in the thousands, and less common organs such as the pancreas, the bowel, and composite tissue, for example, the hand or the face, are also transplanted to help people who have end-stage organ disease. We owe this success largely to pioneers in surgery, science, and medicine, such as Dr. Starzl, after whom the Institute is named. But the truth remains that despite this success, a major challenge is that rejection remains a major problem. Rejection can destroy organs that have been transplanted, and perhaps about 50% of transplanted organs fail after 12 or 15 years after transplantation. So research is needed to overcome the hurdles that stand in the way of solving this problem. And the hurdle, as far as a transplanter is concerned, is the immune system. Humans have highly evolved, sophisticated immune systems that protect us against infection, microbes, but they also protect us against cancer. And unfortunately, this immune system recognizes an organ that is intended to save a life as foreign and will attempt to reject it. And the only thing available to us in our arsenal as physicians and surgeons are immunosuppressive medications that effectively weaken the immune system so that the organ is not rejected. However, they also weaken our defenses against infection, and therefore an organ transplant recipient ends up with complications such as infection, cancer, as well as cardiovascular disease. And we can't solve this problem by simply giving more medications, i.e. solve the problem of rejection, because the more medications we give, although that will reduce rejection, more side effects will be observed. So we need something more magical, more selective. We need to shut off the immune system in a selective way, and that is shown in the green arrow here. We want the immune system to pretend that it does not sense the organ that has been transplanted as foreign and should ignore it, while it continues to protect us against what it's supposed to do, which is infection and other complications. And in order to do that, we at the Starzl Transplantation Institute have taken a comprehensive approach of doing everything from fundamental research to research in humans and a clinical application to achieve this goal. So today you'll listen to presentations that tell you about things we are doing in animal models to things that we're actually doing in humans and how we do them. And when you take a virtual tour of the labs, you're going to see exotic organisms. These are marine invertebrates that live under the sea that can teach us quite a lot about rejection, to fancy microsurgery that we perform in mice to study transplant rejection, and finally, how we study the cells of the of patients that are involved in the rejection process itself. So welcome to this presentation and to this tour of our Transplantation Institute.
Hello, everyone. My name is Geoffrey Camirand. I'm an assistant professor at the University of Pittsburgh in the Department of Surgery and the Department of Immunology. And I'm also a member of the Starzl Transplantation Institute. And today, I will present to you some of our research that we are doing here and focusing specifically on two technological advances that we have trying to develop to help us home in onto specific questions that we have in the laboratory. And so our main goal in the lab is to understand the immune responses that are ongoing inside the transplanted organ. And one specific question that we are after is which cell subset is orchestrating immune rejection within the grafted tissue. There are three main immune cell subsets that are important for immune rejection. One is dendritic cells, shown here in purple, and which can interact with killer cells. These cells go on and destroy the grafted tissue. And there's another subset that is called suppressor cells. So these cells also interact with dendritic cells, and their role is to dampen the immune responses locally. So we, from our previous research, we found that these subsets are highly dynamic. So this is intravital imaging of these immune cells. In blue are the dendritic cells, in green are the suppressor T cells, and in red are the killer T cells. And what you can appreciate is that the blue cells are fairly static. They stay in the same place, while the T cells, the two T cell subsets around the dendritic cells interact. Some have quick interactions and some have more prolonged interactions. And so, so then we ask the questions, are these interactions leading to T cell activation, or are they important for rejection? And to get to this specific question, to be able to specifically visualize T cell activation in real time, we were using calcium sensors that are expressed in these T cells. And this is the first technological advancement that we have implemented in our research that I wanted to talk to you about. And as T cells interact with dendritic cells, the calcium levels, and they get activated, their calcium levels is increased. So with these tools, we can measure specifically when there is a low or a high calcium within individual cells, because there's a change in color, whether it's low calcium or high calcium. So like I mentioned, the goal is to use these calcium sensors to visualize T cell activation in real time. And here are two movies I'm going to show you. Again, the colors are the same. The dendritic cells are in blue and the suppressor or killer cells are in red. And so this is within the transplanted tissue. And you see that some of the cells are moving, while others are more static. So then we can visualize the interactions along with what's happening inside those T cells. And here, if you look at the green and the red colors, which indicate a low to a high level of calcium within those T cells, you see these flashes within those cells as they move around and interact with the dendritic cells. So now we can use this tool to really see which interactions are leading to T cell activation within those T cells. And this is, sorry, I forgot to mention something here. Let me go back. And here I highlighted the two areas with the red circles on the right, which show two specific cells that I picked randomly, where you see these flashes inside those cells. And if we zoom in on those one of those areas, as you can see, there is one cell in particular that has this prolonged interaction, but it doesn't always have those flashes happening inside of the cell itself. Another tool that I wanted to talk to you about is photoactivatable Cre recombinase. And the reason why we're interested in using this is that we want to be able to change gene expression inside immune cells that are infiltrating the graft at a specific time. And the technology behind this is that you have the Cre recombinase expressed in two individual separate parts that stay apart inside the cell. However, once you shine a blue light onto those cells, then it activates the two components, and the change conformation they have now a high affinity, and that activates the Cre recombinase. The Cre recombinase can then go onto the DNA of each individual cell and change gene expression. And this is our setup that we did for just testing three different approaches for photoactivatable Cre recombinases. And what we found is that one of them gave us a high level of activity compared to the other two. So we're currently doing more our experiments to test whether we can activate the Cre recombinase in mice. So a summary of our research, the summary of our research discoveries indicate that dendritic cells are orchestrator of immune responses within transplanted organs. And why is this important is because then we can use these, we can use new therapeutics to target these dendritic cells and help patients and prevent rejection in patients. And also we developed new research tools to perform a spatiotemporal gene editing to further enhance our understanding of transplant rejection. I would like to thank our lab members, our collaborators, and our funding for our research. Well, thank you very much for your attention, and I'm hoping you will enjoy the rest of your tour.
Greetings, ladies and gentlemen. I am Dr. Parmjeet Randhawa. I'm a professor of transplantation pathology at the Thomas C. Starzl Institute. I would like to thank you for joining us today at this whirlwind tour of all the research which goes on in our medical center. This institute is a unique place. It transplants not only organs, but it transplants doctors like me. I have been here since 1987, and I am doing as well as some of the patients who have come here for treatment. So in my first slide, I thought I would show you a picture of this city of Varanasi in India. That's where I'm from. And you can see this city has at least one similarity with Pittsburgh. There's a waterfront. Of course, our downtown is more antique, and the water has some holistic properties which are still to be discovered in the Mon and Allegheny rivers. I would also like to begin by telling you that I'm a pathologist, and the lay people don't always understand what a pathologist does. So I thought I would give you a brief introduction. Pathologists spend their life studying diseases. For example, if we take the example of COVID, which is everywhere in the news, we are the doctors which will ask questions such as, what organs are affected by this virus? Or what determines differences in susceptibility? Why are some people asymptomatic shedders, whereas others develop severe breathlessness and need a respirator? What lab tests can we institute to diagnose this disease early? What are the critical steps in the viral life cycle that we can target to develop good drugs? What tests do we need to monitor the response to any treatment that we initiate? Once the treatment is initiated, how do we know when to stop drug therapy? It can be a clinical evaluation or it can be another lab test. And similarly, we may be developing tests to detect relapse of the disease after it has been treated. And we, as pathologists, do not come into direct contact with patients. Instead, we work in labs such as this one, where typically we have rows and rows of gizmos, which are designed to conduct anatomic, biochemical, and genetic tests on different samples that come to our laboratory. Another function of the pathologist is to examine tissues under the microscope in a histology laboratory And the workhorse in a histology lab is this machine called the microtome, which you see here. And a microtome can cut tissues into slices which are like 1/20,000th of an inch. Because if you make them any thicker, the cells start overlapping and you cannot see the details that you need to diagnose disease. The average turnaround time of a biopsy report is maybe 2 or 3 days. If it's a small urgent biopsy, it can be up to a week if it's a resection specimen. But sometimes a surgeon wants a quicker answer and he calls us right into the operation room. And we do what is called a frozen section. We take the tissue, freeze it right there and cut it and give an opinion to the surgeon so that he can modify his treatment in terms of what we told him. And one such biopsy that we might see in this setting is shown here. This patient had an allograft, or rather a transplanted kidney, which was not working well. So when we looked at it under the microscope, we saw this inflammation, and we also saw some enlarged cells, which in the nucleus, it appeared there were granular particles which might have been viruses. However, light microscopy, the maximum magnification you can see is about 1,000, and viruses are smaller than that. So in such situations, we pathologists, you do electron microscopy, where the magnification is up to 100,000 times. And under electron microscope, in the same biopsy, we could see this cell was choked with viral particles inside the cytoplasm, as well as lining the surface of this cell. Now, the resolution of an electron microscope can be further improved if we develop molecular methods to diagnose disease. And the principle of developing these methods is that we extract DNA from the tissue samples, and we shatter that DNA into smaller fragments. Then we sequence those fragments. And to identify the DNA sequence of these fragments, we map them to a part of the human genome. And this mapping results in what we call molecular signatures, which can distinguish normal tissue from diseased tissue, and also one disease from another. Now, the interpretation of these molecular signatures requires machine learning technology, because the human brain has difficulty seeing patterns when you're looking at the expression of 20,000 or more genes, which have been evaluated from the same biopsy sample. And now here's an example of what we call a molecular fingerprint. To the left, we have 5 transplant patients who were doing well. This is their pattern of gene signature in their kidney biopsies. And to the right, we have 5 different columns for 5 different patients who were rejecting their allograft. So this is the signature of the body's immune system when it's attacking the allograft organ. And these gene signatures help us to make diagnosis on biopsy specimens. That was bulk sequencing. Another technique available in the lab is gene sequencing of single cells. In other words, if we get tissue, we dissociate it into single cells. These single cells are then input into a DNA sequencing machine. And we find that each cell typically has a distinct expression profile, and we can cluster different cells by their gene expression patterns. This is as opposed to bulk analysis, where we examine the whole RNA or DNA, and we get an average gene expression. In an average, the cellular heterogeneity can be masked. So if 2 diseases have differences which are not reflected in the average gene expression of the whole cell mixture, you're not going to find those differences. The advantages of single cell sequencing over bulk sequencing can be explained by taking an example of a polyomavirus infection, which is a relatively common infection which affects the transplanted kidney. So single cell sequencing may help us answer questions such as, why does the virus infect only a certain part of the kidney preferentially, the part we call medulla? Why do some cells in a biopsy remain uninfected while others undergo cell death? Which cell-specific gene signatures can we allow distinction between people who recover rapidly and those who progress to chronic renal failure? And what biochemical pathways can we target to develop drugs that will arrest viral replication? The measurement of gene expression to define molecular signatures can also be performed by a molecular barcode technology. in this technology, which is now available in our medical center, the gene that is to be evaluated is targeted by a probe, which is tagged by these fluorescent molecules. So the expression level of this gene can be measured in terms of a light signal. So these measurements are more accurate, and they can also be free of the amplification bias, which characterizes DNA sequencing measurements. So I will like to end here simply by saying that we are making rapid progress to improve the quality of life for those patients who come to our medical center. Surgeons, nephrologists, hepatologists are constantly brainstorming to see what we can do to advance transplantation medicine to the next level. Thank you for your attention.
Hi, I'm Mindi Styn. I'm the Director for Research Administration and Academic Affairs here at the Transplant Institute, and I'm the Associate Director for Clinical Research. While Geoffrey and Parmjeet gave you excellent examples of basic science and translational science going on here at the Institute, I'm going to take a little different approach and try to give you a bird's eye view of the clinical research program that we have going on. And I'll talk about a few of our studies as we go along. I want to orient you to the structure of the Institute, first of all. So we have STI research, and Dr. Lakkis, who you met earlier, is the scientific director. And this is where our basic science happens. So our bench work, our animal research happens through STI research, and we're closely affiliated with the University of Pittsburgh. STI clinical is what most people think of when they think of the Transplant Institute, the surgery and the associated clinical care that goes along with it. And Dr. Abby Humar is the clinical director. So in the past, when an investigator wanted to do clinical research at the Institute, it could be handled very differently depending on whether that research was initiated through STI research or STI clinical. And as we got more and more into translational research, and as our basic scientists were bringing their discoveries into the clinic, we realized that's not the best way to support our investigators. And so we came up with the STI Clinical Research Program. And the goal of this clinical research program is to support investigators and allow them to focus on the science of the research that they're doing, while we take care of the logistics and we provide an infrastructure to support their work. We are fortunate at the Institute to have the resources to develop a strong infrastructure to support our faculty. The STI Data Warehouse was rolled out in 2019 after 4 years of planning. David McMichael developed the warehouse and maintains it currently. Clinical data in the electronic medical record is extremely valuable for clinical investigators. It helps them plan studies, it helps them observe trends in the transplant population. However, these records weren't made for research, and they're actually housed in multiple different electronic medical record databases. And so what the data warehouse does is it pulls all of this information together and makes it usable for our clinical investigators. We also have a single clinical research coordinator team now. As you can imagine, clinical research is subject to a substantial amount of research oversight and a lot of paperwork. So we have a regulatory coordinator who is dedicated to staying on top of the regulations and making sure that we're compliant. We have 4 nurses who take care of our interventional trials. And we have a biorepository coordinator and a visiting fellow who assists our biorepository coordinator in coordinating that study. On the topic of the STI sample biorepository, Camila Macedo Booth is currently the director of the biorepository. It was started in 2012. And our goal has been to recruit recipients prior to transplant and to obtain samples at regular intervals through 5 years post-transplant. As you can imagine, this saves investigators a lot of time and a lot of effort when they're exploring new ideas, particularly if they want to see, let's say, something in the blood pre-transplant might predict outcomes 3 years later or 5 years later. Without this resource available to them, they would have to recruit participants in, collect that blood, and then wait 5 years to see the outcomes. Now with this resource, they can just ask for, say, 5 blood samples from patients who rejected, 5 blood samples from patients who didn't reject, and they can look at those within a matter of weeks or months. And this gives them the data that they need to then apply for competitive funding. As you can see, our biorepository is very active. We have over 1,300 participants. This includes both recipients and donors. They represent over 1,200 transplants, and we have nearly 5,000 samples. Clinical research at the STI has grown significantly in the past 5 years. We currently have 18 active studies going on, and I'm only counting studies that do what we call primary data collection. So these are studies that would recruit participants in and interact with them to collect data. I'm not counting studies that use our biorepository or that use existing clinical data. You can see in 2015, we were heavily dependent on industry funding. Now we have quite a few more investigator-initiated studies, so studies that our own faculty have written and gotten funding for. And it's pretty much equal, our funding from the NIH and industry. And we also have some competitive UPMC funding in there as well. And again, we've really become a lot more active. We were following less than 100 study participants in 2015, and now it's over 200 participants that our nurses are actively following. And this obviously would exclude our biorepository that has over 1,000 participants in it. So I want to give you some highlights of a few studies as I end my talk. As you know, there's an organ shortage. And in the past, when a person had hepatitis C, their organs could not be used for transplant. However, now with the advent of effective hepatitis C treatment, we can actually transplant a hepatitis C positive organ into a hepatitis C negative recipient, and then treat that hepatitis C that the recipient would get from the organ. And so this is a big shift in standard of care over the past few years. And some sites have implemented this simply as standard of care, but we felt that it was important to systematically collect data on the outcomes from this change in care. Those of you who attended Research That Matters 2 years ago may recall Dr. Angus Thompson's talk about his donor-derived dendritic cell research. Dr. Thompson is one of our basic scientists who has been able to translate his discoveries into the clinic. He's partnered with Dr. Humar and Dr. Tivar to be able to do that. So the basic idea here is that dendritic cells from the donor are infused into recipients to modify the immune response. In our DC-Reg liver study, infusion takes place 1 week prior to transplantation, and then those participants, we start weaning off of immunosuppression 1 year later. In our delayed study, the infusion takes place 1 to 3 years post-transplant, and then weaning of immunosuppression starts 1 week later. In our DC-Reg kidney study, similar to our original liver study, the infusion takes place 1 week prior to transplantation, but we don't do any weaning in our kidney study. And then just to round things out and give you some idea of how diverse some of our clinical studies are, I want to bring up these two. The first is an observational study of patients who have undergone a TIPIAT procedure. This is a complicated procedure, but basically the pancreas is removed. The islet cells from that pancreas are then transplanted back into the patient. So it's their own islet cells. It's considered auto transplantation because the islet cells are so important in preventing diabetes. We currently have 6 subjects enrolled, and this study includes both pediatric and adult patients. And we use questionnaire data and blood samples to look at their outcomes. And finally, we have a study that isn't recruiting transplant patients at all, but in the future could be quite valuable for transplant patients. So we know that older recipients are less likely to reject their solid organ transplants, but the mechanism behind that isn't clear. If we could figure that out, we might be able to come up with novel treatments or come up with a novel test that can predict who's going to do better and who might be at higher risk of rejecting. So this is a very first step at looking at that hypothesis. So it's looking at differences in immune cells between older and younger participants who are recruited from the community. Finally, I just want to thank all of our research study participants and our generous contributors who make this work possible. Thank you for joining us, and I hope you enjoy your tour.
Hi, my name is Matt Nicotra. I'm an assistant professor here at the Starzl Transplant Institute. Welcome. You're probably wondering why we're here standing in a room full of fish tanks when we're supposed to be talking about transplantation. And so I'm going to explain to you a little bit about something that's pretty innovative that we're doing here at the Starzl Transplant Institute, which is to use an evolutionary approach to try to understand and discover new mechanisms for how transplants are recognized and rejected in the human body. So specifically, what my lab works on is an organism called Hydractinia, which you probably haven't heard of before, but it's similar to like a coral or a sea anemone in that it lives in the ocean on a surface. And as it grows, it covers its surface and eventually it bumps into other members of its species. And when it does that, it can recognize whether they're related or unrelated. And if they're related, they fuse together and become a larger animal. And if they're not related, they start fighting with each other until one of them kills the other one off. Now, if you think about it, that's sort of like what happens in a transplant, right? You get an organ from another member of your species, and your body recognizes that as not part of you and starts to reject it. So what we want to understand is whether what's happening in these animals, which do essentially natural transplantation experiments, has anything to do with what's going on in the vertebrate immune system. So these animals here, I'll take one out of the tank so you can take a look at it. We grow them on glass microscope slides. And they just live attached like this. And as they get bigger, they'll bump into each other. And under the microscope later, I can show you what that actually looks like under high zoom. And so what we're trying to figure out is how they do this. How do they recognize whether they're related or not when they don't have the kinds of the parts of the immune system we normally think about with in the human body. So in the human body, you probably have heard of HLA molecules, which differ between people, and that's what's recognized by your B cells and your T cells, and that's what triggers an immune response. So in humans, B cells and T cells use proteins on their surface that signal through different pathways, and those you can think of those as essentially things that tell the cells whether they should attack or not attack. What we found in Hydractinia is that these organisms use other proteins that on the surface don't look like the proteins that B cells and T cells use. But inside the cell, the part of that protein that sticks inside the cell has some things about it that look very similar to the parts of the B cell and T cells that tell those cells whether to be activated or inhibited, or whether to reject or not reject. And so what we're in the process of doing now is understanding how those pathways work in Hydractinia, because that could point us in new directions for thinking about how those pathways work in humans as well.
My name is Martin Oberbarnscheidt. I'm an assistant professor at the Starzl Transplantation Institute. And today I would like to show you some of our research. Behind me is a 2-photon intravital microscope that is a microscope that uses pulsed laser light to excite fluorescent proteins in cells that will emit light so that we can visualize different cells in a live animal studying their cell interactions. What I would like to do now is demonstrate some of the image data on the computer screen to you. The main focus of our research is on how transplanted organs get rejected by the immune system of the body. And specifically, we're interested in how one of the major players of organ rejection, the T cell, is getting into the transplanted organ. And to answer that question, we employed intravital imaging of transplanted mouse kidneys to answer that question. What I'm showing you here is an image of a transplanted mouse kidney showing in red the blood-filled capillaries of the kidney, and in green cells that surround those capillaries. And these cells are tissue-resident dendritic cells. These are cells that live in the kidney that are able to pick up foreign antigen and present it on their cell surface to other immune cells in the body. And the microscope here allows us to record a 3-dimensional image volume that can be rendered by the software and rotated in many directions. What helps us in analyzing these image volumes is that we can create surfaces according to the signal that we're recording with a microscope. And now you can see smooth green dendritic cells surrounding these capillaries in the kidney. One of the first observations in a native kidney was that these dendritic cells are not only surrounding the capillaries, but they're also inside of those capillaries. And that is demonstrated in a different movie that I'm going to show you. So what you can see here is kind of a fly inside of the capillary, and you can see these green processes from the specialized antigen-presenting cells that surround the capillaries inside of them. This was an important observation that we followed up in kidney transplants. This movie is demonstrating a kidney that was transplanted 3 days ago, and what you can see here are blood flow and blood capillaries shown in cyan. You can see T cells of the recipient, the cells that can mediate organ rejection, in red. And you can say the specialized antigen presenting cells, the dendritic cells of the donor kidney, in yellow. And you can see the same cells of the recipient in green. And what you can appreciate is that there aren't that many T cells around that can destroy the organ, and that there are mainly yellow cells, which are the dendritic cells that came with the kidney graft and are of the donor origin. Now, when you look on day 7 after transplantation, you can see that the picture has changed dramatically. Here again, you can see the blood capillaries and the blood flow in cyan, and you can see the dendritic cells of the recipient in green. And as you can see, they are highly outnumbering any potentially left cells in yellow of the donor, indicating that the dendritic cells of the donor left the kidney, and now we have a massive population of the transplanted organ with the green recipient dendritic cells. That was one of the first studies that demonstrated that dendritic cells of the recipient go to the transplanted kidney and then infiltrate it. And we then asked the question, how do the T cells actually get into the graft? Now, one point I would like to remark is that these dendritic cells have their origin in monocytes that circulate through the blood. And one of the other observations that we made is that these monocytes are initially infiltrating the organ, and then they differentiate into these specialized antigen-presenting cells that are shown here in green. Now, if you look at the T cells, in this video, we again have blood flow marked in cyan, and we have dendritic cells of the recipient here shown in green, and we have T cells of the recipient that are infiltrating the graft. And we made the observation that these T cells are in the graft tissue now, and they are closely interacting with these green cells. They're touching each other, and they're staying in place here. Now, if you take a closer look, sometimes you can catch T cells that are in the process of leaving the blood vessel and migrating into the kidney to cause their rejection there. And what you can see in this movie is that there is a red T cell inside of the capillary that is leaving it while interacting with that green recipient dendritic cells. And further experiments have shown that these dendritic cells of the recipient that reach inside the blood vessel are extremely important to mediate the T cells infiltration of the organ and leaving the blood vessel and getting into the tissue. And the tissue is the place where these T cells can now mediate their effector function and destroy the graft and cause graft rejection. So overall, our research for the first time has shown that it is dendritic cells that present foreign antigen from the graft to the T cells of the recipient to mediate their infiltration of the graft and to enable them to cause graft rejection. This important step in investigating how T cells get to the graft and mediate graft rejection will hopefully enable us in the future to find ways to interrupt this process and allow the T cells to not get into the organ and cause graft rejection. And with this research, we hope in the future to be able to improve patient outcomes and improve lives after transplantation. Thank you very much.
Welcome to the Microsurgery Suite. This room represents one of the pillars of the Institute. Without this room, we wouldn't be able to do our basic science research, which involves transplanting rodent hearts and kidneys. As you can see, this work is very difficult and requires the use of microscope. This specific microscope is a teaching microscope where the teacher and the student can view the same field. The skill is very difficult to master. It takes a novice anywhere from 3 to 6 months to be able to become proficient at it. That is because some of the vessels are very small, only millimeters wide. And the sutures that are used are thinner than your hair. So it's amazingly difficult to perform this task. This room also has very specialized equipment, such as the anesthesia machine, to make sure that the mice are kept under for the duration of the procedure, as well as heating pads, since mice lose body heat very quickly, and as well as buckets where we can keep the mice and keep them from running away. Now, we wanted you to get an idea of what it is to transplant a mouse organ. Dr. Dai here has harvested a heart organ from a mouse. You can see that the organ itself is very tiny, and just without a microscope, you will not be able to see the vessels themselves. This is essentially one of the most difficult skills that you can master here at the Transplant Institute, and it's essential to our work. The University of Pittsburgh was where some of the basic principles of microsurgery was developed by the father of microsurgery, Dr. Sun Lee, and this excellent work is continued on today by people like Dr. Day. We hope that you have enjoyed this tour and that we will see you in person very soon.
My name is Dr. Christopher Hughes. I'm the surgical director for liver transplantation at UPMC. And we're studying in our lab a process called sonothrombolysis to try to increase the effectiveness of liver transplantation and prevent some of the complications after liver transplantation. One of the main problems that we have in dealing with donated livers is the fact that there may be thrombi, or what is small blood clots inside the liver that are never cleared before the liver is transplanted, and those can damage the liver later on. And so there's been a problem within the liver transplant world as to how do we get rid of those clots, and how do we make it so that when we reperfuse new blood to the new liver, the liver is able to survive well and do well. And so our project that we have is to use a tiny bubble called a microbubble that's infused by the billions into the liver in a machine outside the body before the liver's transplanted. And those tiny bubbles circulate through all the veins and arteries in the liver. And then if those bubbles are submitted to an ultrasound field from outside the liver, those bubbles will explode. Billions of tiny little explosions inside the liver that basically break up any blood clot that's inside. And so we do this process over a period of a few hours before the liver's transplanted to be able to clear the liver so that when we do transplant that liver and restart blood supply to that new liver, we don't have problems with areas of the liver which aren't receiving blood because the blood clots being there. And so this is a sort of a mechanistic approach to causing tiny explosions in the liver to clear it out so that the recipient can have a nice liver which is healthy after it's transplanted. And so our project here right now, we have a rat liver that's being perfused artificially. It's alive, it's working, it's making bile and performing all of its functions. And once we have the perfusion like this going, we'll submit the microbubbles into that perfusion and then the ultrasound to clear that liver of any blood clots. And so once that procedure is completed, we'll take the liver off the machine and we'll transplant the liver into another rat to see how it does and see how it survives. And once we finish the rat studies, we'll move on to human studies and test the human liver on a much bigger scale, but with the same tiny microbubbles to see the effect there. And we're hoping that in the long run, it will improve the outcomes for liver transplant overall.
Hi, my name is Camila both. I'm a physician scientist here at the STI. I came here 20 years ago for a postdoc fellowship and never left. Now I'm a junior faculty. I'm going to be showing you today this instrument. This is a flow cytometry cell sorter that has different functionalities. And again, over this last 20 years, this evolved in a very different way. What we do, I'm going to show you here, like you have your mix of cells, your whole blood, your drop of whole blood. Then you add some antibodies, and then these cells would pass through this fluidic system, and then by lasers, they're going to identify each cell what it is. If it is in a white blood cell, it is a T cell, a B cell, NK cell. And these all happen on these machines. Here we have, this instrument has 5 lasers. We can go up to 30 colors, so 30 different functionalities of these cell types. And and what we can do in terms of experiment-wise, we did in the past, we co-culture cells from a transplant recipient with its donor and put in a culture for 5 days. And after 5 days, we look at how they react to each other, how the recipient was reacting to the donor cells and peripheral blood. So then you can see each dot here represents one cell, and you can see how they proliferate, how they go against each other. So this way give us an idea how the recipient is reacting against the donor. And we can also look at functionality of the cells, not only cell types, but how functional they are. So this instrument definitely, it was a breakthrough through the whole immunology community, especially in the transplant field. What else we can do, it can sort cells. What it means, again, the same thing. You have a mixture of cells, you add antibodies, then you pass through this instrument, and according to what cell type you want, it is going to different containers. So you can put each individual cell in a different tube. So this way you can have those individual cells and look for different things. As we did it, for example, on this experiment, we have recipients were infused with donor cells for an immunotherapy, and we could track, according to the HLA of that between the recipient and donor, we could track those cells in peripheral blood. So after the cells passed through the sorter, we sorted the dendritic cells, what we call, and then we could see, for example, in yellow here are the donor cells, and in blue, the recipient cells. So then, and this is the cell type that we want to see. So then they do have different fluorochromes. That's how the lasers pick up the signal. So that's how we can divide them and know exactly what they are. And then we can see the interaction between the recipient and the donor. Then the donor is picking up, the recipient is picking up some particles of the donor, and then can give a regulation, an immune regulation, so, or the opposite, and we can try to track if what is happening is going to reject or not, because of we could see each individual cell visualize and see the functionality of that. And this, again, these come from this instrument that has these two main capabilities. There's a sort of sorting the cells or just looking at what specificity of the cells. We have about 3 instruments like that and in the core that do the sorting, and we have 5 that can do the basic flow cytometry. It's very user-friendly. You do your own sample. So each researcher here, each postdoc, fellow, they can do their own experiment, and it goes as far as they want in terms of science. So nowadays, I would say this is definitely one of the good signs of the good lines of science. So I hope that I could explain a little bit.
Welcome all of you to Oberbarnscheidt lab. I'm a research instructor and I work with Professor Martin Oberbarnscheidt. But a little background, I'm a medical doctor who came here to do research, specifically basic research into transplantation immunology, specifically into the intragraft immune responses, meaning what happens inside the graft after transplantation. How cells get in, what do they do in there, and how do they perpetuate damage. And with that line of work, I delved into single-cell RNA sequencing, where we gathered a lot of data about how genetically cells are doing, what they're doing in the graft, basically damaging it. And for that end, to be able to study and to really understand these cells, I leverage artificial intelligence, and I use multiple machine learning algorithms to be able to truly understand how these cells are at the transcriptomic or genetic level are working. And the cells in question here are cells that I've been studying for the past 3 years, are the tissue-resident memory T cells. So T cells, when you have the transplant, T cells get activated in something we call secondary lymphoid organs, such as the spleen or the lymph node. Now, after that activation, they migrate into the graft. And I focused on after how they migrate on the graft, and I looked at the T cells when they get into the graft and then persist there. How do they change and how do they do what they do? And what we did is we utilized specifically a kidney transplantation model where the kidney is foreign to the recipient. We then transferred cells that are specific to the kidney, and it specifically damaged that kidney. And then we harvested the kidneys and got those cells after week 1 or week 5 post transfer. And we also took out heterogeneous population of polyclonal cells that can also invade the graft. And we then did single-cell RNA sequencing, where we have this awesome machine that can detect all the genes that are being expressed and how much they are being expressed in the cells that are being sequenced. And as you'd expect, when we sequencing, we're getting around 12,000 cells. That's a lot of cells. And we're getting gene information about these cells, and these genes we can detect up to 15,000 genes. Now that results in something called highly dimensional data. And that highly dimensional data requires sophisticated algorithms to analyze. And one of the algorithms that I utilize is called UMAP, Uniform Manifold Approximation Projection. And what this algorithm does, it tries to understand how highly dimensional data is situated in highly dimensional space, and then it projects that understanding into a lower dimension. As you know, we human beings, we can only see or understand in 3 dimensions. Machines can translate this highly dimensional data based on, and specifically here, all the cells with all the genetic expression, and then project it in 3D so that we can appreciate patterns. And the pattern that I saw in these very antigen-specific cells, the OT1 cells, over time from week 1 to week 5 is that they lie on a spectrum. So you have week 1 cells lie on one end of the spectrum, and then week 5 on the right-hand side here, the other end of the spectrum. And then utilized another algorithm called pseudotime analysis, where it can then project how, with pseudotime, these cells are differentiating. I use k-means clustering, another algorithm that can partition the cells into different time points along that trajectory. And then when I looked at specifically a transcription profile or transcriptional program associated with tissue-resident memory T cells, my cells are adopting that program as they're upregulating genes specific for tissue-resident memory T cells, and downregulating a gene specific also for tissue-resident memory formation. And we're also able to look into other cells, not just the antigen-specific cells that I transferred. And we have polyclonal cells also. And now we have gene information about them. We know how they are situated in higher dimensional space. We use classification algorithms to know by genetic what cell types they are, and then we can analyze them separately. And for the sake of my own work, I looked on one neighborhood or cluster of cells, and its genetic profile was also associated with memory. Now, with all of these data, we're not only detecting cell types, cell clusters, and we're not only looking at how the gene transcription is changing over time, but now we can look at how certain clusters have genes that are shared with other clusters. And more importantly, we can employ multiple techniques. Specifically here, I use ingenuity pathway analysis that can look at gene networks that have already been studied and defined. And in the subsets that I was interested in, I was able to detect that there is a activation or inhibition of certain pathways specific for my tissue-resident memory T cells, which allows me more to understand their function. And more importantly, we can now look at different properties of these genes and see if they are important regulators of the cells in question. And those genes now, through this network analysis, we can look at the whole pathway of genetic expression, how they affect other genes, and if that gene can be targeted. And that's by compiling all the information that we got from the dimensionality reduction, the pseudo-time series expression analysis, and the differential gene expression analysis. All of this data with the network analysis is combined, and now we can detect gene targets. And specifically in my analysis, when I looked at all of the data, there was one specific transcription factor called BHLHE40, which sadly, when I detected it to be an important gene, using all that information, one day prior to that discovery, I saw that somebody published on it that it is actually associated with tissue-resident memory T cells, but that didn't let me down. It actually confirmed that what I was doing is right. And I went on and started looking into this gene list, into other genes that could be important, and then I landed another gene that might be important. And I looked back at the data and was able to see that this might be a gene of importance, and now currently I'm focusing on it, trying to understand its role with these cells and the function, and hopefully being able to target it so that specifically we can remove the cells that express that gene, and these cells would be harmful. So by targeting that gene expression of that specific cell, we can then remove the cell in question. So here the main question that I am focused on is delving into all of this gene information that we're getting on these cells, and then trying to find a specific gene that I can target. And as you can see, that's the whole layout of what I'm doing, but now I'm trying to scale it up, basically trying to build a curated gene information database. So for every gene, for these cell subtypes, I'm trying to gather as much information as I can and then provide a label, whether it is targetable or whether it is a gene of interest or not. And I want to make that database accessible to all researchers so that that can help their research as much as helping mine. And to do that, I will be doing exactly what I was doing before with the pipeline, getting single-cell RNA-seq data, cluster the data, doing differential gene expression analysis, classification, and employing different algorithms to gather more information about these genes. I'm also concurrently with this manually outputted pipeline, I'm also trying to automate the whole process with collaboration from people from the Department of Bioinformatics. We automate the whole process, and then we try to employ a neural network that can, by itself, through our own coding, And through training on data sets that we have and other data sets, it will provide the label itself, whether this gene is a target or not. Well, as you can see, they saved the best for last, and this concludes your lab tour. And I can send you off now to Professor Lakkis, the director of the Institute.
In closing, I would like to thank everyone for attending this virtual event and the virtual tours of the labs. I hope that you've really enjoyed it, but I also hope that you recognize the breadth and the depth of research that we do here at the Starzl Transplantation Institute at the University of Pittsburgh and UPMC. Our mission, our goal is to improve the lives of transplant recipients through both fundamental and applied research. Again, I would like to thank you for attending, and I wish you the best.